This is the coronavirus. Here I've got my coronavirus that I made. It's a sphere, and it is really not this big, right? It's about 1,000 times smaller than the diameter of human hair. So you cannot see it. And the interesting thing when you think about this virus is it is not an animal. It can't move on its own. It doesn't have any wings or legs. It traveled around the world fairly quickly, though, because humans transmitted it. So we'll talk a little bit in lecture about how what the theory is, one major theory of how this virus jumped from animals to humans. I think I already did mention that in the nutrition chapter, actually, that it believed to have originated from bats and then transferred into pangolins, which then made its way into humans in a mix of humans and live animals in a wet market in Wuhan. So that's the major theory, is that this was transmitted. And going back to this is, how does this virus, how did it manage to take over the world? Well, it is picked up off of surfaces. This can survive on surfaces, and it is transmitted from a host. The host that is suffering from the virus is producing the virus, and then basically has an output of the virus either through a cough or a sneeze, which is the reason for masks. And when someone who's suffering from the virus has a cough or a sneeze, it aerosolizes the virus and sends it out into this vicinity. And this virus cannot last that long outside of the host. And so that's the interesting thing, is that the early days of the virus, and even continuing now, especially with other variants, it is important to know that we can fight this without any fancy vaccines or medications, simply by realizing that this transmits from person to person through basically not realizing that you're carrying a virus that you might have picked up off a surface from your hand, and then transmitting it onto your face, which then makes its way into your body that way. So your hand becomes a vector. And why? Why does the hand become a vector? You don't realize you're picking this up because this virus, this white part of this model, it has an envelope that is a fatty membrane. So we have fatty membranes in all of our cells of our body. But when I say fatty, I mean fats, like fats and oils, those structures of fats and oils that we're going to review in this lab. There's this is fat structure in this white outer part of the membrane. And we know one thing about fats and oils is that they're attracted together. So this fatty outer membrane of the coronavirus is attracted to oils on your skin that you are naturally secreting. And so this is how this becomes aerosolized, lands on a surface. On that surface, which could be like a cup or just a table, this can last not that long, but in that short amount of time, if you touch that, then it goes onto your hand, not unintentionally, because oils on your hand are attracted to the fatty membrane, and it sticks there. And then when you touch your face without a mask, there you go. So the other thing you'll notice about this structure is these black things that are sticking out of it, right? So these black things that are sticking out are instrumental in how the vaccine was developed. So let me pull one of these out. Let me find one that I can remove here. This is called a spike protein. So the virus has about 90 of these proteins sticking out of there, and it has a very specific structure. And so one thing that's important about so first of all, why it's called coronavirus in the first place is because of those spike proteins that they radiate out from the sphere like a corona, like the sun, like a crown. And so corona is Latin for crown. And so that's how this in part got its name. There are 7 coronaviruses that are known to cause illness in humans, including SARS, which was first really began to be studied more than 15 years ago, the SARS virus. And so the thing about this coronavirus is, because it was one of the family of the SARS virus, scientists could quickly understand and develop vaccines faster than any time in human history. Most vaccine development took anywhere from 10 to 50 years. Well, this took about 1 year to develop the vaccine. Now, there's fear about that. Is it real? Can we trust it? And the answer is yes, because when you really think about it, the science behind the coronavirus and the science behind the coronavirus vaccine goes back more than 15 years. And so it is something we're going to get into in the module this week and next week, understanding how this vaccine works. It is all part of natural systems in our human body, and it is not unnatural. It's not being injected with any kind of craziness from, the government. And so this is part of this, going back to this protein. There are 90 of these proteins, and these are the spike structure. This structure is what latches into a receptor in your body once this virus makes its way into your body. So this is basically like a docking system where this gets on your hand, the virus makes it in, and then the spike proteins play an important role in really incorporating the entire virus into a cell. So this spike protein attaches onto a receptor that's naturally present in your human body. And that is called the ACE receptor, and it's in your lungs, it's in your heart, it's in your GI tract. And this is why the virus will attack the heart and the lungs and the GI tract. And what it does is it leads to sickness, because when this spike protein docks and one of those receptors in your lungs or your heart or your GI tract, this attaches and it docks and it enters that cell, the heart or the lungs or the GI tract. And what it does is it takes over the cell. So let's say your heart cell is just doing its thing. It has its function as a heart cell. Once this virus is incorporated in, it causes that heart cell to go berserk and not do what it used to do, which is act like a heart cell. And so there you've lost the function of the heart cell. And what instead that heart cell does is it starts to reproduce and create more of this virus. That's what viruses do, is they fool your body into making more of the virus and take over any cells in your body. And the goal is for the virus wants to propagate itself. And so if that is in your heart cell, or more importantly, in your lung cells will start to make a lot of the virus. And then in the lungs, the virus will then be excreted, expelled through a cough or sneeze onto the next host that picks it up. So that is the human host immune system can fight off the virus. But that's where the differences arise, where some people have pre-existing conditions. Some immune systems are known to go a little bit overboard and lead to multi-organ failure. So there are different reasons for the fatalities of the coronavirus. And each person that gets the coronavirus has unfortunately, a different result. Okay, so we're getting into the coronavirus and the science of soap, how soap is important in fighting off this virus. So first of all, make sure you have the lab procedure and lab report handy. So let me show you here on my screen. Okay, so here we have the lab page. So the lab procedure and lab report is one page. So I'll show you this. Okay, so we have the background, which is I'll go into the science of this reaction to make soap. And I'll do that on my slides. But there are going to be videos that I'm going to stop and show the videos and give you time to answer these questions. And also I'm going to review on my slides this reaction with making soap, and which also will review fats and oils and structures of saturated fats and unsaturated fats. So we're going to get through all of this. So have this lab report ready, and you should be able to complete it during lab. So actively working on this. Okay, so let me see. I am going to first my slides here. Okay, so we know that Soap is important in washing your hands, washing your body, and fighting the virus. If you've ever tried to take a shower without shampoo or soap, just using water, it doesn't work, does it? That's because your body is producing oils naturally, whether in your hair or on your skin, and those oils do not wash off with water. Soap is a middleman that connects the oils on your body, on your skin and your hair. It connects those oils to the water and allows it to get washed off. So the other thing that soap also does is it can break down this virus. And so that's twofold there. You get clean and you're killing things at the same time. So exactly how soap works against the coronavirus, we'll get to in this video. But first you have this structure here. And remember, this is your spike protein, that's the red. And those outer spike proteins are important once the virus is already introduced into your body. So how is it already introduced in your body? That's that first part. This is the fatty outer membrane. And that's the white part. And it's gray on here. But that fatty outer membrane is attracted to oils on your skin. So let's go to the first video. This is coming from the name of the virus, coronavirus, and originating in the year 2019. So that's coronavirus disease, or that's the VID part in 2019 is COVID-19, same thing as coronavirus. So let's talk about soap. So this is the structure of the soap molecule. And the soap molecule is a similar structure to fat. So this is similar to a fatty acid, and we have seen that fatty acid structure before. The fatty acid has a tail. This is a nonpolar tail. And what I mean by nonpolar is, remember, the nonpolar molecule has all nonpolar bonds. And we can see that the atoms in that tail, the bonds are carbon-carbon bonds and carbon-hydrogen bonds. So when you do the electronegativity calculation, those are less than 0.5, and that's nonpolar, and it means that the electrons are evenly shared. So there's an equal distribution of electrons in each of those bonds, so there's no partial positive or partial negative. And so what that means is that you don't have any positive or negative in there, and this nonpolar is only attracted to other nonpolar compounds. Now, there are not many nonpolar compounds, but one that's very common is fats and oils. Okay, so this part of the soap molecule is attracted to fats and oils secreted on your skin or in your hair. And that is very similar to the structure of a fatty acid. There's the same nonpolar end. Now, that is called hydrophobic. Hydrophobic means it's afraid of water. And so that's what fats and oils are, is they are the exact opposite of water as far as attractions. And so this is the hydrophobic end, the nonpolar tail, and it basically will be attracted to fats and oils on your skin, but also want to get away from water. Well, the other part of the soap molecule is kind of unique. It has a polar end, which is actually more correct as the ionic end. So this ionic end has a negative charge, which I'm highlighting in red, circle that. And then it also has a sodium ion that is a positive charge. So it is attracted to, so the ionic end is attracted to other ions, or more specifically, water. So it's attracted to other polar compounds, and very importantly, it's attracted to water. So that means it is called hydrophilic. Okay, so that's what soap does, is it can attach to oil, and that's oil on your skin. And that oil on your skin once it's stuck to the soap, now has the other end of it, this side that is the ionic end, that will then attach to water. So the soap is a middleman, and it allows for the oil to be pulled off of your skin and get washed off with the water. So that is similar to a fatty acid, the structure of the soap, but not exactly the same, because the fatty acid has a carboxylic acid at the end. And so this is the carboxylic acid group is in short COOH, or it has the structure that looks like this, the double bond oxygen, single bond OH. So you see that's very similar to the soap molecule that has the double bond oxygen and single bond oxygen with a negative charge. So why is that so similar? It's very similar. What the soap molecule does not have is a hydrogen, but it does have this ionic end, the negative charge and the positive charge. And why is that so similar? It's because the soap molecule originates from a fatty acid starting point. Okay, so that's a fatty acid reactant in the reactants. So the way soap works, though, is it's called a surfactant. It can clean off surfaces. So it can clean a surface like your skin, or it can clean, fabric surface. It can clean a table surface. And the way it works is that this is the soap molecule. This is the zigzag line is the nonpolar tail. That's the nonpolar end. And then this is the ionic end. Okay, and so that's what these soap molecules look like. See how there's that zigzag line? It's not because it's a little bacteria thing that's moving. That is exactly the shape that you had seen before. This shape, it has a zigzag line that is the nonpolar end. That's the line angle drawing. And then this is the ionic end that has the positive and negative. So that's a negative and a positive. I'll draw that here like this, cartoon style. So there's a positive and a negative. Okay, and so what happens to the soap is that the nonpolar end, that zigzag line part, will attract to oils. So it will attract to and also attach to other nonpolar compounds like oil, fat, dirt, coronavirus membrane. Those are all nonpolar compounds. Okay, and so this is the example of dirt. So the nonpolar tail, the squiggly part, is all what's attracted to that dirt. And what happens is that the soap molecules will surround whatever the particle is, whether it's oil or fat or dirt or a virus. Okay, and in that process of surrounding, we've seen before that it's similar to how water, the process of water dissolving an ionic compound. So what water does is it will be attracted to the ions in an ionic compound and surround the ions and pull them off one at a time. We've seen this a few times in lecture and in lab. I showed you that video. And that process is this is what soap does. It's just that soap is a nonpolar end that's doing the surrounding. So the soap molecules surround this dirt and literally pull it off of the surface. Just like the water can dissolve an ionic compound by literally pulling off the ions one at a time. And so once this is surrounded, this will remove that particle from whatever the surface is. Now it has to be, the particle originally has to be a nonpolar particle. So the oil, the fat, dirt, the virus membrane, it has to be nonpolar for the soap to work. Okay, but as long as you have some water incorporated in, now this part, the polar end, is attracted to water, and this will get washed off. So this will wash away with water because all this outer end, the outside of it now is what's attracted to water. That's the polar end or the ionic end. And when water comes along, water will then take this with it, take this whole package with it. Okay, so that's how soap cleans surfaces. Now, in the science of soap, we know that the virus is not only washed off with soap. So here's the same soap molecules. These soap molecules have that same interesting looking structure. This is the polar end, and that's the non-polar end, that squiggly line. So remember, the virus has that fatty outer membrane, and that fatty outer membrane is non-polar. So notice what is the virus will attach to skin because it is also non-polar. There are oils on the skin, as well as the skin membrane cells are also non-polar. So non-polar is attracted to non-polar, and so the virus will stick to oils in the skin or the skin itself. So what happens though is the soap molecules are attracted to the virus because that non-polar end, that squiggly line, notice is what is attracted to and attaches to the virus there, just like that. The non-polar end goes in and attracts to the virus and surrounds that virus. So cleaning off the surface, just like the dirt worked on that previous slide, is that the soap molecules surround the virus. So the soap molecules surround the virus and pull it off. So surround the virus particles and pull it off of the surface, whether it is your skin or the surface that you're cleaning, could be a table. And that's why soap is important when cleaning a surface, not just using only water for your skin, because that's not going to do it. It's not going to clean off the oils. It's not going to clean off the virus, or not just using water or just wiping off a table, but actually using soap. Because that will not, soap is needed to pick up the dirt, the bacteria, the virus particles as well, and any oil too, if you actually want to have a clean table. So let's take a look at the next video here. Let me go to here. Is the soap, how soap is made. So how soap is made is called the saponification reaction. This is called the making of soap. And this is a reaction that goes back centuries. So it goes back to the days when humans made everything. Basically, you'd kill an animal, you'd eat the meat, you'd use the skin for making, a fur coat, or, leather for shoes or bags. And then you'd use the fat from an animal, but the fat can be an animal or a vegetable source or plant source. And so we know that in general, fats can be, animals provide fats like butter, milk fat from cows, or oils tend to be from plants like olive oil from olive trees. And then lye. So lye is this sodium hydroxide, NaOH. It's called lye. This is the old-fashioned name for, basically before the understanding of chemistry, is that you could get this ash from a mountain and mix it together with the fat from the animal you killed. And so this fat you should recognize is called a triglyceride structure. So this triglyceride has 3 fatty acids, and those 3 fatty acids are 1, 2, 3 right there on a glycerol backbone. So the glycerol is the 3 carbons with 3 OH groups. We saw this triglyceride structure in module 11, and triglycerides are in foods. And so the 3 fatty acids can be all exactly the same fatty acid, or they can be different fatty acids. And on this, when you have a triglyceride from food, This can be from an animal or plant source. And then you mix it with lye, the saponification that happens is something that takes time. It's because when you have a fat, this sodium hydroxide is an aqueous solution, which means it is in water. It's dissolved in water. Well, when you have something dissolved in water, trying to mix something with a fat and oil does not mix with water. And so because fat and oil does not mix with water, the saponification reaction has to be done over a long period of time. You have to stir it and then stir it some more in the process of making soap. So soap making was a process that would take the whole day. And if we were on campus, we'd be making soap. It takes about an hour of stirring, a whole hour of stirring. Okay. And why is that? It's because fat and water, so the fat and the water do not mix. And so what happens though is when you finally get it to mix in the slow reaction, you will release the glycerol. So this is the glycerol. It's 3 carbons, 1, 2, 3 with 3 OH groups. And you will also have, this is the soap structure, the same structure that we saw before. There are the carbons that are the nonpolar tail. So drawing the line angle drawing, and then the carbon double bond oxygen, single bond oxygen with a negative charge, and then the sodium positive charge. So that has the nonpolar end and the ionic end, okay? And so this is the soap molecule. This is the soap molecule, and the glycerol is also mixed in there, okay? And so what happens is that fats and oils, we know have the general properties of greasy, slippery, soft, low melting, and not soluble, generally solids. Oil and water don't mix because this is a fatty acid. Stearic acid is an example of a fatty acid. So let's draw this structure again. This would be converting this to the line angle drawing. So this has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Here's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 carbons. So this is called the line angle drawing. Remember, when you convert the carbons to the ends of the line or any place where 2 lines intersect. And then on this end, this is called the carboxylic acid end. The carboxylic acid, converting that into the carbon with a double bond oxygen, single bond OH. This is the OH group. Okay. So this is the fatty acid. It's very similar to the structure of the soap molecule. So the soap molecule has that hydrocarbon tail, but instead of having the carboxylic acid, it has that ionic end. And that's because it originates from the fatty acid from the fat. Okay, but it's slightly different because the sodium hydroxide in the reaction adds the sodium on that end. Okay, so it's very similar to the fatty acid, the soap structure. So fats and oils, we know, are attracted to other fats and oils, but they're not attracted to water. And why they're attracted to other nonpolar molecules is because nonpolar molecules are attracted to other nonpolar molecules. They're all basically avoiding water. They're hydrophobic. So that's why the soap molecule is different and allows for hydrophobic, the nonpolar end, to attract to the polar end, the ionic end, which will then go into water being hydrophilic. Okay, and so there are fats and oils. So the fatty acid we just saw is part of a triglyceride, and oils also can be part of triglycerides, and these are all part of the lipid family. So the main difference between fats and oils is either you can say the animal source or plant source, or you can say that fats generally are solid at room temperature and oils are liquid at room temperature. But they all belong to the lipid family. And that's one thing to point out here is that cell membranes have phospholipids. That falls into the part of other lipids. So let's add phospholipids. And that's phospholipids. It's called phospholipid. That's an H. that has lipid in the name, and that's in cell membranes, are part of the lipid family. They are fatty, and same as this outer membrane of this virus, this coronavirus, that white part of the coronavirus that I'm showing you on my screen, that also has phospholipids. And so that's why this is the non-polar part, is attracted to non-polar fats, non-polar oils, and other cell membranes like in your skin. So this is the triglyceride, is when you have 3 fatty acids and glycerol, and triglycerides are present in the food you eat. Now, when you add 3 fatty acids onto a glycerol, those fatty acids can be saturated or unsaturated fatty acids. So this is a review of the monounsaturated and polyunsaturated and unsaturated fats. So this is when you have one double bond. It's called a monounsaturated, one double bond. Saturated, let's actually write that here, is a saturated fat. That's what I had previously drawn, is a saturated fatty acid, has no double bonds and all single bonds. And the reason why it's called saturated is because it has the maximum number of hydrogens. It is saturated with hydrogens. That's why it's called a saturated fatty acid. Once you incorporate one or more double bonds in there, you no longer have the maximum number of hydrogens, and that's why it's called unsaturated once there's a double bond in there. So having one double bond is monounsaturated, one double bond, compared to polyunsaturated, which can be 2 or 3 double bonds. Now, this linoleic and linolenic acid, both of these are examples of what are called essential fats. So these are essential fats, and we talked about these in lecture, that these are essential to human metabolism. Your body needs them, but your body cannot create them. So you need to get these through your food. Okay, so the examples, actually, let me go back to need to be eaten. The examples we looked at of essential fats can be had in higher amounts in these superfoods like avocado, olive oil, and salmon, that have the polyunsaturated fatty acids, because in general, these come from plant sources, and plants can produce polyunsaturated fatty acids. Humans cannot produce polyunsaturated fatty acids as well as plants can. So when you have a double bond, that double bond can have an arrangement that is either called a cis arrangement or a trans arrangement. So in the cis arrangement of the double bond, the hydrogens are on one side. So drawing in the hydrogens, they are on one side of the double bond, and the result is an overall bent shape. See how that's kind of a C shape there. So that bent shape can't stack up. When you can't stack these things up, this becomes a liquid at room temperature. And so when you have a liquid at room temperature, that's an oil, example of an oil, because it flows, and is also considered a healthy fat because it doesn't stack up in your arteries. And this compares to the trans fat. So this is a trans fat, which has the hydrogens on the opposite sides of the double bond. And this results in an overall straight shape. And that overall straight shape can stack up. And when they stack up, this becomes a solid at room temperature. And these trans fats are generally going to be called fats like butter or coconut oil. And these are unhealthy fats. And they are unhealthy fats because they can stack up in your arteries and lead to plaques and blockages, so leading to heart disease and stroke. Okay, so the fatty acids, when you have 3 fatty acids on the glycerol, that makes the triglyceride. And so this is the glycerol molecule, has 3 carbons with 3 OH groups. And then when you add on 3 fatty acids, so these are your 3 fatty acids, they are all exactly the same, but they don't have to be exactly the same. But these 3 are exactly the same. attached to the glycerol backbone, you have a more complicated molecule. And so this is called your triglyceride. Okay, and this is a triglyceride in food. Now, most foods have 3 different fatty acids. And so that's why we saw that in lecture, is that foods have a combination of fatty acids when you break them down. There's going to be monounsaturated, saturated, polyunsaturated in different percentages. So you have different percentages of the fats in foods, or different percentages of the fatty acids in foods. Some are unsaturated, some are polyunsaturated, some are saturated. Okay. So let's actually, one more thing I wanted to make sure, hold on, let me go back to here, is in this molecule, I think this one. Okay, so this is the soap molecule. And one thing about the soap molecule is, how is it attracted to water, that hydrophilic end? So let's add the water molecule. Okay, so the water molecule is H2O. And remember, it has that bent shape. So that bent shape has the V shape like this. And the water molecule is polar. So it is polar. And it has the partial negative end is the oxygen, and the partial positive end are those hydrogens. Okay, and so let's draw this more just like a V like this. Okay, so the negative end is the pointed end, and the positive end of the polar water molecule is the open part of the V. So how the water molecules arrange is on this soap molecules, it's not attracted to the nonpolar tail, but the water molecules are attracted in a very specific purposeful way, that the negative ends of the water molecules are attracted to the positive end of that soap molecule, or the positive ends of the water molecule are attracted to, let me make a little space here. Okay, the positive ends of that soap molecule are the negative of the water, and the positive ends of the water are attracted to, so the positive end is open, the open end are attracted to the negative end of the soap molecule. And so that's the water molecules will then be attracted to the soap, and the soap is already attached to the oil, this part. And so this connection between the water and the oil happens through the soap molecule, because it has the ability to attract to both nonpolar and polar. Okay, so you've got, let's see, I think this should get you through everything on page 3 here. Line angle drawings, water soluble and fat soluble portions of soap. Okay, so let's go to here. Nope, don't want to do that. Don't want to do that. Okay, all right, let's go back to here. So we are going to talk about now, look at one last thing, which is how soap does become important in the process of killing this virus, and how the hand washing is really something we can all do. So let's take